Hi everyone. Today we are going to look upon how the topics genetics and molecular biology can be connected. So let's get into the topic. So, what do you mean by genetics? Or what is genetics? Genetics is the branch of biology that deals with the study of heredity and variation in living organisms. So, what is meant by heredity and variation? Heredity is the process in which how the traits or the characteristics of organisms are passed from their parents to their offspring, and variation is the differences seen among the persons of the same species. So, genetics is the combination of both. How the heredity and variation is happening in living organism can be studied with the help of the branch of biology known as genetics. Okay, now we can just examine this picture. What can we see in this picture? We can see a human being out there, and we know that a human being or any kind of body is made up of millions and trillions of cells. So, if we examine one such cell out there, a cell consists of many cell organelles. We know that nucleus, mitochondria, uh, etc. There are many types of cell organelles out there. So, among that, nucleus is the most important cell organelle, isn't it? So, inside that nucleus, we can see thread-like structures known as chromosomes. And if we examine that chromosome clearly, we can see that these chromosomes are made up of DNA. So, and this DNA is constitutes of nucleotides. And what do you mean by gene? We have told about chromosomes, DNA, and nucleotides. So, what do you mean by gene? Gene is a segment of DNA. Most probably, we used to have a confusion regarding these two terms, what is gene and what is DNA. So, gene is a segment of DNA. Okay. So, inside a cell, we can see the nucleus, and the nucleus consists of thread like structures called chromosome, which are really the DNA molecules. And these DNA constitutes the gene segments, and these genes are made up of nucleotides. Okay, so now we can look upon the structure of DNA. As we all know, DNA is the double helical structure. The double helical model of DNA was proposed by Watson and Crick, and this DNA consists of many nucleotides. And what is special about these nucleotides? These nucleotides consist of sugar, phosphate, and nitrogen bases. And if the sugar molecule is deoxyribose, then it is deoxyribose nucleic acid or the DNA. And if the sugar is a ribose sugar, then it will be a RNA, ribose nucleic acid. Okay, so in DNA, the sugar molecule is deoxyribose, and it also consists of phosphate and the nitrogen base. So as you can see in the topmost picture, a nucleoside is one which constitutes the sugar and the nitrogen base. That means if the sugar and nitrogen base is together called as a nucleoside, and if a phosphate group is added to that structure, it becomes a nucleotide. So a nucleotide consists of sugar, phosphate, and nitrogen base, and a nucleoside consists of a nitrogen base and sugar. So if a phosphate group is added to that nucleoside, it will become a nucleotide. Okay, then we have told that nucleotide consists of nitrogen bases, and these nitrogen bases are mainly of two types, that is purines and pyrimidines. What are purines? Which are the major purine molecules? That is adenine and guanine. And what about the pyrimidines? Thymine and cytosine are the major pyrimidines. So these purines and pyrimidines are attached together by the base pairing rule. That is, the adenine always pairs with the thymine with a double bond, and guanine always pairs with cytosine by triple bonds. These purines and pyrimidines are attached together by hydrogen bonds, and the adenine always pairs with thymine by two hydrogen bonds, while hydrogen bonds are also seen in between the guanine and the cytosine, and the guanine and the cytosine are attached by three hydrogen bonds. Hope you got the point. And we have already told that DNA is a double helical structure, means it consists of two strands which are anti-parallel in nature. That is, one will be 5' to 3' and other will be 3' to 5'. So that's all about the structure of DNA. The structure of DNA, or the double helical model of DNA, was proposed by Watson and Crick, and the nucleotides are the major components of the DNA. And this nucleotide consists of sugar, phosphate, and nitrogen bases. Nitrogen bases are mainly of two types, purines and pyrimidines. These purines are the adenine and the guanine, and the pyrimidines are thymine and cytosine. And adenine always pairs with the thymine, and guanine always pairs with cytosine. And the structure of DNA, we can also see that two strands are anti-parallel in nature. Next, we can talk about the packaging of DNA. As we have seen in the previous picture that the DNA is packed inside the nucleus, isn't it? So, as you all know that even the nucleus is a very small structure, so DNA have to be fit inside the tiny nucleus. So, it must be packed very carefully. So human DNA is very long. It is approximately about 2 meters of length in each cell. So it must be fit inside the tiny nucleus, so it should be packed carefully. That's why the DNA packaging is done with the help of proteins known as histones. So what are histone proteins? These are positively charged proteins, and we all know that DNA is negatively charged. Why DNA is negatively charged? Because it consists of phosphate groups. So DNA is negatively charged while the histones are positively charged molecules. Why the histones are rich in lysine and arginine, which led to the formation of the positively charged molecules known as histones. So they also help in neutralizing the negatively charged DNA. So that's why the histone and the DNA are fit in tight manner, because histones are positively charged and DNA is negatively charged. So they help in each other by neutralizing the negative charge of the DNA. Next, we are going to discuss about the five main types of histones, that is H1, H2A, H2B, and H3 and H4. The last four are the core histones, that is the H2A, H2B, H3 and H4 are the core proteins, and they are 2 in number each. That constitutes a total of 8 proteins. These 8 proteins form a core, and they are known as the core histones, and the rest H1 act as a linker protein which helps in further compaction. Okay, so this DNA wrap around this histone core molecules, they form the structure known as the nucleosome. So with the 8 proteins, 8 histone proteins attached to the DNA, they form the structure known as the nucleosome, as you can see in the picture in the right side. So we have told that there are 5 main types of histones, and they are 2 in number each. So total there are 4 into 2, 8 proteins will be there. These 8 proteins are attached to DNA by the linker protein known as H1. So the wholesome product is known as the nucleosome. Next, we can see in detail in the picture, we can see the nucleosome, where it is formed of DNA and the histone core, while these nucleosomes are further coiled to form the chromatin fiber, and this chromatin only condenses to form chromosome during the cell division only. So what is said here? The nucleosome is formed of DNA and the histone core, while the chromatin can be seen as a further coiled form of the nucleosome. The nucleosome further coils to form the chromatin fiber. That is the main structure, or that is how the chromosomes are embedded inside the nucleus in the normal way. But when during the cell division, this chromatin condenses to form the chromosome for the cell division. Okay. So nucleosome, then the nucleosome leads to chromatin fiber, and this chromatin fiber later becomes the chromosome during the cell division. Hope you got the point. Next we can say about the two types of chromatin, that is euchromatin and heterochromatin. As you can see in the picture itself, heterochromatin is tightly packed. So if they are tightly packed, they will be in their inactive form. So it consists of inactive DNA, and they are transcriptionally inactive regions. While you can see in the euchromatin region, these contain loosely packed and active DNA, and they are generally expressed genes. So euchromatin are transcriptionally active region of the chromatin. So euchromatin are the loosely packed and heterochromatin are the tightly packed. Next, we are going to talk about the central dogma of molecular biology. What do you mean by central dogma of molecular biology? It means how a DNA results in the formation or expression of a protein, or it says about the characteristics or the phenotypic characteristics of an organism, how DNA results in a phenotype of an organism. So for that we need to know about certain processes. So what do you mean by replication? If a DNA is formed from another DNA, that process is known as DNA replication, or simply the replication process. A DNA is formed from DNA. And if an RNA is formed from a DNA, it is known as transcription. And if a protein is formed from RNA, it is known as translation. So this protein results in the expression of a phenotype. So from the DNA, RNA is formed. From the RNA, a protein is formed, and protein results in the expression of a phenotype. So replication, what will be the product of replication? DNA will be the product of replication. And what will be the product of transcription? And RNA will be the product of transcription. What about the product of translation then? The protein will be the product of translation. Okay, that's all for today. So we have just talked about how genetics can be correlated with the subject of molecular biology. Hope you all understood. If you have any doubts regarding the session, please comment about them in the comment box. Okay everyone, thank you so much for listening. See you in the next class. Bye.